Things moving across the membrane today. So, how things move, how things get in and out of your cells is a super important concept. It helps you to maintain your homeostasis and to be able to stay alive. Two vocabulary words that I super need you to know are those polar and nonpolar words. When we talked about those in the last unit, it was just kind of that water is a polar molecule. Okay, thumbs up. That's pretty much all you need to know. Now I really need you to know the difference between a polar molecule and a nonpolar, because one of these is able to cross the membrane quite easily and the other one requires a little bit of assistance. So, easiest way to think about this is symmetry. Is the molecule perfectly symmetrical? If the molecule is perfectly symmetrical, then it is nonpolar. No one molecule is exerting itself on the other one. Polar is non-symmetrical. One of the molecules is exerting pressure or pull or itself on the other molecules that are a part of it, of the other atoms that are part of that. The way that it exerts its pull on the other atoms that are going to be in that molecule is due to the valence electrons. And it really goes back to this larger concept, if you remember from your chemistry, called electronegativity. So an uneven amount of electronegativity, one from the right side of the periodic table and one from the left, when they go together, they're probably not going to be balanced and then pull properly or nicely on one another. Sometimes they do. Often they are not, because you have multiple things that are trying to connect themselves together. So polar, not symmetrical; nonpolar, symmetrical. If you need more help, if you need extra things, if everything I've said for polar versus nonpolar is just not sticking in your head, please watch this crash course video. So hit the pause button right now, go take yourself over to the slide deck, watch the crash course just on polar and nonpolar. You don't even have to go to the slide deck. You can literally just go to YouTube, type in polar versus nonpolar crash course, and it should pop up right away. And then I would recommend that you watch that video if you need just one more little piece to help you to really understand the difference between polar and nonpolar. There are three ways that things are going to get across the membrane. And really, there's just two ways that things move and are transported across the membrane. This third one is kind of involved in all the rest of them, but it's really different, so we kind of separate it out on its own. The three different ways and the three things that I need you to know are passive transport, active transport, and then this thing called bulk transport. So in transport, typically the difference between active and passive has to do with where are you moving in relation to the concentration gradient. And the concentration gradient just says, well, where is the highest density of molecules and where is the lowest density of molecules around things. Think of this concentration gradient. I think the easiest way to think of this is in terms of cooking or a candle. If you have your candle and you light it, or you start cooking some food and it is in your oven or on the stovetop, generally the smell just stays right in that kitchen, right in that area. But the longer you wait, the further and further away that scent, those volatile organic compounds, will waft. So they're going to move from that area of high concentration to low. Sometimes, though, we need to move from low to high. It's naturally normal for molecules to move from high to low. They want to have their own elbow room. They want things to be spread out and evenly spaced. Sometimes we need to reverse that. So we'll get to that as we go. First type, passive diffusion. We're going to start with just your regular plain old diffusion. These are just small nonpolar molecules moving across the membrane. Think of things like oxygen gas or nitrogen gas or carbon dioxide. These are going to be really little molecules that are able to move across the membrane freely. They don't get tangled in the phospholipid tails. They slip in between those hydrophilic heads, and they can just get in and out as they need. These guys are most interested in creating equilibrium. We need them to be able to get out and in at a pretty efficient rate. So luckily, they're tiny. They just get to do their own thing and they can move across. It does not take any effort on the cell's part to let these guys in or out. In fact, we need them to move in and out so quickly that it is in your best interest to not require any energy for these guys to move across the phospholipid bilayer. So no energy, little tiny things, always from high to low concentration. This is the same thing that you were supposed to have identified in Model 2. It is one of the questions off of that. You were noticing that those dots are the only things that move in and outside of the cell. Those are those small non-polar molecules. Sometimes polar molecules, if they're small enough, will also be able to slip through. Generally, though, it's just your non-polar ones, but there are a couple polars that are, if they're tiny enough and they don't have a lot of polarity to them, they also will just be able to slip through. Small is going to be the key on this one. Small, relatively small, no charge to these guys. So why diffusion works, how it happens, is it's just a space issue. It's just a random movement of things. You know this, solid molecules are packed pretty tightly together. Liquid molecules, they're a little more fluid. There's a little bit of space in between those guys. And then gas molecules have a tremendous amount of space in between them. So because gas molecules have so much space in between them, also the space in between these guys is based on the amount of energy that each molecule has within itself. They're going to bump into that phospholipid bilayer at a pretty high rate. Because they have so much energy in them, because they are bumping into the phospholipid bilayer so often, that allows them to just move freely across that phospholipid bilayer. That doesn't work for everybody. Small nonpolar, usually your gas molecules, are so high in energy and so tiny that they bump into the phospholipid bilayer so frequently, they just get to move themselves all the way through. 4 things that impact how quickly things move across that membrane. Diffusion can happen in a wide variety of substances. It can happen in the atmosphere. It can happen in gas. So like think about sound or smell, like we talked about. It can happen in water. You've all put a drop of food coloring in water and then watched it diffuse from there. It also happens in liquids within your body and in all organisms' bodies within their cell. The 4 things that make that difference you can see on the left-hand side there. And the way that it works is temperature, like we just talked about. The more temperature, the higher the temperature, the more collisions, the faster the diffusion will happen. The other 3 factors are also intuitive. We're going to spell them out here for you. So the first one is going to be pressure. Think of the soda. Any carbonated beverage that you have, if you make that carbonated beverage, you shake it up a little bit or you get it nice and warm, when you crack that soda can or you twist the lid off a plastic bottle, it has that psh noise. That's a whole bunch of carbon dioxide gas diffusing out of the liquid under pressure. So it's been held under pressure. That's why if you squeeze on the walls of a warm soda or a cold soda, it's better if it's a cold soda. And then if you squeeze on a can, you'll notice that the walls are pretty rigid. That's 1, because yes, the walls are rigid, but 2, because the carbon dioxide that is in that liquid is under a tremendous amount of pressure. So that's why as soon as you crack the pop tap on the top of a fizzy water even, or a soda, or you do it to the plastic bottle of, again, either fizzy water or soda, it has that psh, because all the carbon dioxide is coming out because there was high pressure. Concentration gradient is also going to be a factor. All 3 of these first 3, so the top one and then this one we're going to talk about now, are exactly the same as they are with an enzyme. So you can see the difference here between example number 1. If we have 10 carbon dioxide molecules on the outside, 2 on the inside, versus example number 2, where we have 4 on the outside and then 2 on the inside of the cell, which one's going to diffuse faster? It's going to be cell with the 10 on the outside. It's going to try to push those to the inside at a faster rate than the 4 into 2, because it's just trying to achieve that equilibrium at a faster rate. There's more on the outside, more chances for collisions. We're going to get those guys on the inside. So cell 1 will have the greater concentration gradient, going to diffuse fastest. Molecular size is going to be your last factor. If the molecule that's trying to move across that phospholipid bilayer is tiny, it's going to be able to move across more easily. The bigger they are, the higher just even a little bit of polarity on there slows everything down. So you want things to be really little, really warm, high amount of pressure, big desire to move from one space to the other, and then that's going to make diffusion happen faster. And then all of those in reverse would slow diffusion down. The second one is facilitated diffusion. And this is, again, same as diffusion. We are going to be talking about typically small molecules. But now we can't just move through the phospholipid bilayer. Walking through walls is no longer an option. There's something about the molecule that is trying to cross the phospholipid bilayer that just needs a little bit of assistance. Typically, these are just going to be like a door. You can think of it like a screen door, but not a screen door where you have to like actively slide the door out of the way per se. These are more like a screen door where you can just like run through the screen, and it has those cool magnets that when you run through it, they just go whoop and let you through. And then when you're done going through, they go click, 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 click. And then they just go back into place all at once. Or you can think of them like ribbons that are hanging down across the door frame. There's like a little bit of resistance to having anything and everything. It's not just a wide open gap, but there's not really anything that's preventing a whole lot of stuff from moving. It's totally accessible, absolutely usable. Very little energy is required in this case. It's kind of just the energy to put that protein channel in there. But aside from that, there's no energy. We're still moving from a high to a low concentration. Now we just have a little bit of a specialized space that we can move across that particular spot. These are going to be a 1-to-1 ratio, so one molecule to one protein channel. And usually it's because there is a specific molecule that has a specific charge, or the polarity is a little bit high, or the molecule is just not quite right that it can slip through that phospholipid bilayer. So it gets its own special door, and one molecule to one door, that's it. Some molecules will also need some sort of a transport mechanism. They need to be like scooped up in a little basket and then hand carried all the way across. And you can see that in these two different facilitated diffusion depictions. One, it looks like just kind of an open channel, and then the other one has to do a little like open, close, open on the other end movement to get those molecules across the membrane. Those channel proteins technically have two different names. The one that's just like a wide open channel, like a Suez Canal or the Panama Canal or something, like it's just open. You can just like doop, 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 go all the way through. On the other side, the carrier protein, we actually have to like hand move you through the membrane. You cannot just get all the way through on your own. You need assistance all the way through. Think of, we have to stick a tugboat in front of your ship in order to carry you all the way through that channel that we're trying to get you through. Typically, channel proteins are for ions. Really, ions don't need a lot. It's just that ions, because they carry that charge, they get tangled in the fatty acid tails. So having a little space for them is the one that we'll get into, that'll allow them to get through. On both of these, typically just your carrier protein though, that is what Model 3 was trying to show you. Sometimes to get the channel to open, and typically to get the carrier protein Model 1 to open, we need to bind some sort of a hormone up here to get the door to open so that we can use that particular channel. You'll notice in Model 2, I'll take you back to that guy, you did not really have the difference between a carrier and a channel protein. So just know that it's not as simple as just two little like bars that are holding something open like a door frame. There's a little bit of stuff that's in there to help facilitate things getting out and things getting in. We also do not want the wrong stuff to get out, or the wrong stuff to get out, or things to just get out willy-nilly in the cell. Everything is tightly regulated. These guys just make life possible for particular molecules as we need them to move in and out of the cell. The type of channel that is made has to do with the folding of the amino acids in the polypeptide chain. So whatever we need to move in and out of the cell, however, whatever channel I need made, the cell will code for that specific amino acid chain, and then things will get folded up specifically. I'll show you the picture here in a second. Again, it's a 1-to-1, so every channel protein or carrier protein is unique for the molecule that needs to get in and out. This is very similar to the way that it was one enzyme to one substrate. There's only one lock for your one key for your front door lock, so that's the only way that you can get into your house. You can't go take your key to your friend's house. And the last thing on here is that gated idea. Some of these, this does not apply to everybody. We don't all need that. Not all channel proteins, not all carrier proteins need those hormones, need that gate in order to help open them. Sometimes they do, though. We'll specify what they are and when they are. You don't have any specific examples that you need to memorize right now. I just need you to know that that is the case. So model 2 is a thing, and then model 3 is sometimes supersedes that channel protein, and there's just a little extra piece that has to happen on there. Water and your osmosis lab that you have either done or will be doing. Osmosis is not something that is big and fancy and extra. It's just a specific type of diffusion, and it is literally just the diffusion of water. There's really nothing special about this. There's nothing unique. There's nothing different. It's just diffusion of water. So it's a facilitated transport. There is a specific channel protein that has to happen because water, as you know, is a polar molecule. So we have to have a special spot for it. It doesn't take any energy. It follows all the rules of regular diffusion, high concentration to low concentration, moving that stuff across. So nothing that's special there. The name of this particular channel protein is aquaporin. That's right. For the first time in biology, we named something exactly like it sounds. Aqua pours in, water pours in, aquaporin. That's all that this is. It's a pretty nice name that we can remember that one. I do need you to know this channel protein's name. It's the only one that you have to know at all. Aquaporin is for water. It's kind of in the title. Before we can go any further, we need to talk about 3 types of solutions. And when we talk about these 3 things, they're all going to be relative. So we're going to compare the inside to the outside. Everything is relative. What applies to one set of scenarios does not apply everywhere. It's much like ski hills, I recently learned. Each green, blue, and black style of runs at a particular ski hill is dependent on that hill specifically. There's no standard for greens across the world. There's no standard for blue style of ski hills across the world, and there's no standard for blacks. It's all relative to that specific mountain. Here's what these 3 are. So we're going to start in the middle. Isotonic is like an isosceles triangle. You have equal sides, or in this case of the cell, you have equal parts. Stuff on the inside is the same as the stuff on the outside, and what we're talking about when we say stuff is the concentration of molecules inside to outside is the same. In the hypotonic situation, I think of hypo like hippo. That helps me to remember. There are going to be more solutes that are on the inside of the cell than on the outside. What that does is it makes sure that the cell is nice and full. Sometimes that's a good thing. As you're watching this little GIF down here underneath hypotonic, sometimes it's unfortunate and it has some unintended consequences. Flip to the other side of this slide and look at the hyper kid or the hypertonic scenario. You'll notice that kid is going bananas. He is just super excited about something. And these cells that are down below are also doing the same thing. They are super excited. They're going bananas. You can see they're moving really fast across the cell. And what we commonly associate with hyper people is they typically are very thin. They're very small. You can see that these cells in this picture are also kind of small. They don't look round. They don't look circular. They're not nice. They're shrivelly and shrinky. And I'll show you some more pictures here in a second. We typically do not want cells in a hypertonic state. That's not good. Animal and plant cells want different things. So hypotonic is not good for the animal cell. It generally means exploded cells. Exploded cells are not good for animals. That's bad. That typically means we have to replace that cell. That costs us energy. We don't like that. Plant cells, on the other hand, big fan. Big fan. They get that vacuole nice and full. It's all plump and has water in it. And so that vacuole onto the cell membrane, onto the cell wall can exert pressure, and it can help that cell to stand up nice and tall. We are not talking here in plant cells about wood, so please don't hear me with trees. Trees are like kind of a whole separate, different thing. Leave those guys out. This is going to be your vegetables, your flowers, your non-woody stemmed plants. So think of anything that's in that realm. Plants want hypotonic. Nobody likes hypertonic. Hypertonic is sad. We have words for hypertonic. I'll give you those in a second, but just keep this in mind. I'm hoping these gifts are like helping to create a little silly image in your head, but ain't nobody like the hypertonic state. It's no bueno. This is the difference between hypo and hypertonic for a plant. Okay, you want to be the big one. You want hypotonic things. Hyper does not work for a plant. These are like your sad. It's either time to start watering the heck out of that bad boy, or just let it go. You done messed up. You didn't water it enough. All those cells are hypertonic. A hypertonic cell will eventually be a dead cell. So water your plants, please. Last but not least, we have the isotonic situation. This is the one that's for animal cells. They're a big fan. This keeps them full and happy. They want the inside and the outside to be the same. Plants, on the other hand, they pretty much go hypertonic when they do not have enough water. Hypotonic is the only way for a plant. Isotonic is the only way for an animal. What happens in that plant cell is not enough pressure is from the vacuole to the cell, to the cytoplasm, to the cell membrane, to the cell wall. And so this is your plant that's not falling over, but it looks sad. It's sad and droopy. Those plant cells are in an isotonic state. So take a pause button here, and I want you to think about which direction is the water going to move in these situations. Is it going to move into the cell or out of the cell? Always the amount of the solution has to be 100%. And the solution is made of all of the solutes plus all of the solvent. So if you have the solutes, that's the stuff, that's like the Kool-Aid powder or the hot chocolate powder or any powdered anything that you are trying to make. The rest of the solution needs to be water. So hit the pause button now. Each one of these, inside, needs to add up to 100, outside needs to add up to 100. And I want you to consider and think, okay, which way is the water going to move? How do I get this generic cell to have a proper movement of water into the cell or out of the cell? Hit the pause button, quiz yourself. In the first scenario, you should have gotten 99% water, 1% solute, 97 to 3. That means there's more water and fewer solutes on the outside than on the inside. So we're going to get that water moving to the inside to try to give some space to those solutes. For the second scenario, you should have gotten 95 and 97 again. And what that should tell you is that there is a greater amount of solute to water ratio on the inside. So the reverse happens. We're going to move that out, and we're going to try to give the extracellular solution a little bit more space, the extracellular solutes, a little more space between them. And then on the bottom one, hopefully you got 97 and 97 and you thought to yourself, well, she probably gave us one of everything. This one's probably the one where it's happy, it's isotonic, things are moving in and out just properly. Couple vocabulary words for you and just the difference in nice charts. These would be good to print off if you wanted as blanks and then work on filling them in so that you really do know that information. And then remember, you also want to be able to apply the information to scenarios. So I want you to think about how is the water moving in the isotonic situation? How is the water moving in the hypertonic situation? And how is the water moving in the hypotonic situation? And then what's the end result going to be for the cell? Hit the pause button right now. See if you can't figure those out. And then I'll go through them with you. So in the case of the isotonic, hopefully you figured out, okay, inside, outside, iso, same. Simple. Water in, water out, no, totally normal. Thumbs up for an animal cell. On an hypertonic, hopefully you remembered that this is water out pretty much all the time. Those cells are so stinking excited. They're just giving themselves up all the time. They're just moving around. Everything is going crazy in there. So we're taking the water outside. Typically there's more solutes outside, so that's why the cells are giving up their contents. They're really just giving up their water. And this means that those animal cells will shrivel up. And then in the last state, you should have gotten that those cells are getting filled up with water. So then all of a sudden they burst. New vocab word for you here is this word in parentheses called cytolysis. So lysis, just like you learned from hydrolysis to split. This one is cytolysis, so cytoplasm lyses or explodes. So exploding cytoplasm. It's no bueno. We do not want that. Flip it over to an animal cell. 3 vocab words are still the same. What we are doing here in this isotonic situation, you can hit the pause button, quiz yourself, but you also have the same pictures to the side. Just going to give you some vocab words that are here because these work the same way. The first one, in an isotonic state, those cells actually become, and the plant becomes flaccid. In the hypertonic state, when water is rushing out, what we have is we have a concept called plasmolysis. So we have plasmolysis. The plasma membrane or the cellular membrane is lysing. It's shrinking away from the walls and eventually it rips off of those walls and then it shrinks down to nothing and then that cell dies. So that's no bueno. And then in the last case, hypotonic, we love these types of cells in our plants because they have a high amount of what we call turgor pressure. So that is the name that we give the concept of the vacuole pushing on the cytoplasm, pushing on the cell membrane, pushing on the cell wall. And it lets that plant sit up nice and tall. Give yourself a little quiz. Hit the pause button again. How does a cell rid itself of carbon dioxide? I've given you 2 concepts, passive diffusion and just regular diffusion. Sorry, facilitated diffusion and passive diffusion. Which one is it? Go back. CO2 is on one of the previous slides. I'm not giving it to you right now. How carefully did you read those slides? These are all 3 of these together. So aquaporins are out there separately on their own, but we have channel proteins, we have carrier proteins, and then we have that passive diffusion or simple diffusion. It's also said that way. So just like this diagram, because it puts all 3 of them on the same page, so you can just look at everything I've just been talking about all together. The next one we're going to talk about is active transport, and that is what Model 4 was trying to show you, is that active transport is very different from the either simple or facilitated diffusion styles. We got to move against the concentration gradient. When we move against the concentration gradient, typically this is going to require some ATP. We're going to need to have some specialized transport proteins in this case. We call those specialized transport proteins pumps. So we're pumping stuff against the concentration gradient. Nobody likes to be a salmon swimming upstream, but sometimes you got to suck it up and you just have to do it anyways. So that is what happens here with this active transport type of movement. ATP, as we're going to learn later, that is cell energy, and it costs some cell energy, some cell cash. You can think of ATP like cash. It costs a little bit of energy to be able to do this. So typically the ATP is what gets spent in order to make this happen. This is generally used for small polar molecules. There's just some times when you need to move things across the concentration gradient in the wrong direction from low to high. It just has to happen sometimes. 3 examples for you of times when we need that to happen are this one called the sodium-potassium pump. Then there is an electrogenic pump. And then there's a thing called a proton pump. We are going to get into the proton pump later. You can see it pictured here in a different use than we are going to talk about it. The electrogenic pump is also interesting. And then the sodium potassium pump is the reason and way that your nervous system gets to function. So this diagram right here comes out of your textbook, and it's a pretty great one. You can read these 6 things, but I'm going to recommend that you hit the pause button here and go check out the active transport mechanism for the sodium potassium pump. It's how and why your nervous system functions the way you do. If you took anatomy and physiology, you may have already learned about the sodium potassium pump. Unless you are an expert, I would recommend everybody go watch the sodium potassium pump video so you understand one super, super critical functionality of an active transport mechanism. Sometimes it takes more than one pump to get things working. You can see that in this diagram. We sometimes have a molecule that we call a co-transporter. To get sucrose into the cell from the outside to the inside, because it is a larger molecule, we're going to use a proton pump in conjunction with a sucrose transporter, and it becomes this little like cycle of getting the sucrose in. We call that, when it's a double pump situation, co-transport, because cooperate, we're taking 2 pumps to get something to move in the way that we need it to. We got 2 concentration gradients that we got to go. All right, so last but not least is this concept. If you go back and you take a look at your cell structure, you might have a question. I didn't, but I want you to think about this, ponder. How do those protein glyco things get there? How do those happen? Well, it works off of a concept called an active transport style thing, exocytosis. This is for very, very large molecules, bigger than a protein channel can handle. So what'll happen sometimes is after the Golgi apparatus folds up things that need to happen. Remember, the Golgi apparatus is like the UPS of the cell. It will package up the larger molecules, or maybe there might be just like a ton of molecules that we need to get out of the cell at a particular moment, and it wraps them up in a vesicle. It fuses that vesicle with the cell membrane, and then it just becomes one. And then if there are little molecules that need to be released, they get released. If they are little molecules that now need to become part of the cell membrane, like those protein channels, that's how they go become a part of the phospholipid bilayer. So they come from the inside, they're manufactured inside the cell, they are packaged by the Golgi body into a vesicle, and then they fuse and become a part of the phospholipid bilayer. Getting things in is also tricky if we're trying to get something very, very large in, bigger than a protein can handle, and or if we're trying to get a whole lot of something in all at once. Basically, just take exocytosis and reverse it. That is known as this middle one, actually called penocytosis. We're just pulling a whole lot of stuff all the way in. Sometimes when we need to eat large things, like when one cell eats another, as in the case of your immune system, because perhaps that cell was walking around with a Collins ID card, and we were like, excuse me, friend, you don't go here. And then we have to gobble them up and destroy them because they're an invading pathogen. The model, or the name that's given for the immune cells that can do that, is called phagocytosis, where they can literally actually just one cell can gobble up a whole other cell. And that's only for your immune system. The bottom method of endocytosis is when we need to get the cholesterol, your HDL cholesterol, from outside of the cell into the cell, because we need to make it part of your cell membranes. We need it to do some certain things. How we do that is we scoop up that cholesterol molecule, pull it in, and then fuse it into the membrane. So that's how HDL cholesterol, if it's being incorporated in the membrane, that's how it works. If you need more information, if you need more of anything, I'm going to recommend that you watch this crash course video on membranes and transport. It'll give you this great big overview of every single piece that you could possibly need. So please take a moment, go watch that video, and you will learn lots of things that I have just gone over in a shorter amount. He talks real fast. You might want to pause that guy, but you can get a lot of information in a different voice from a different perspective on membranes and transport if you need someone and something else. If you have any questions, if you need formatives reset, please ask. I would be happy to help you.